Okay, folks, so we're going to introduce the microbes chapter. I'm going to get through some of our basic separations to understand, like, how we kind of understand our microbes, and that way we can break into the actual chemistry on Friday. So let's go ahead and get started. I don't have a ton of time. This shouldn't hopefully take the whole class period, so just get to a certain point. But in this chapter, the basic idea, we're looking at how microorganisms will play a role and fit into all the stuff we're learning about, like organic matter and like colloids and doing this. We're going to look into classification and properties of microorganisms. Eventually, we'll get into the role in the degradation process of organic matter and how this affects the potential energy, the doing work by electricity, and the Gibbs free energy. And then we'll look how they affect the carbon, nitrogen, and sulfur cycle. But up until now, we haven't really distinguished between abiotic and biotic processes, like carbon dioxide getting trapped as carbonates and whatnot. We haven't really looked at where the source of carbon's coming from and how degradation of organic matter can be both this way, both just a natural process by weathering and also a process that's brought about by microbes. Because in general, photosynthesis converts inorganic carbonates to organic carbon in our green plants. So our CO3 minus, which is essentially an anion, is converted into an organic compound. Okay, so the major biotic processes that we'll deal with are due to microorganisms. That, much like our aerosols, much like our colloids, these guys can range in size, but they're often very small and frequently in the micrometer, thus the microorganism range. But these guys are found in great number almost everywhere. In fact, if you added up all the total mass of all the microorganisms, it's many times greater than even that of all the large animals put together. So if you measured up all the microorganisms, that weigh more than all of the like blue whales. It's like the thing about ants. There's more ants on Earth than like any other creature, any other at least animal. So when we talk about microorganisms will do a lot of different chemical reactions, such as they can convert ammonium to nitrate, which is a very important process. It uses oxygen and produces some acid. This nitrification is a rather straightforward equation. However, this equation, as simple as it looks, is not a single-step reaction. And without microorganisms, it will not occur to any significant extent in abiotic chemical fashion. In fact, even though Gibbs free energy, which controls spontaneity, which means the likelihood of this to occur, so this is a downhill process, it wants to happen. That's negative 265, so this really wants to happen. However, ammonium is highly stable in air. So even though this wants to happen, there's a large activation energy barrier to break in order for this reaction to start occurring. This reaction, though, in soil and water can be mediated by a class of bacteria called Nitrosomonas and the Nitrobacter bacteria. And so this can happen easily when these bacteria are present, bacterium, but normally not very easily. Many such processes can occur for all types of carbons, nitrogens, phosphorus, sulfurs, even iron and manganese. So we'll be looking at some of these processes in terms of microorganisms, what they're responsible for. So there's a variety of ways to classify our microorganisms. And the first one is the phylum. What family do these live in? There's bacteria, which is the most abundant type of microorganism. They can be found in water and in land, and they range between 0.2 micrometers to 50 micrometers. Bacteria have a very high surface to volume ratio, meaning they're kind of really small and have a lot of surface area. So they are usually deprotonated, which means they carry a negative charge, thus the gram-negative term for most bacteria. And they can be in a variety of different shapes. They can be spheres, ellipsoids, rods, and they can exist in a variety of different conditions. So it all depends on the bacteria themselves, what shapes or what conditions they thrive in. But most bacteria require some amount of nitrogen, phosphorus, potassium, and carbon, which they will incorporate from various organic molecules that they can chew up. Now, the actual population amount of each bacteria depends on such things like the temperature, the pH, the electrolytic concentration, so ionic charge, and of course the nutrient supply and moisture content, especially on land. So if any of these guys is particularly low, we're not going to have a lot of population, but once these build up high, we can grow a lot. However, in general, we see a pretty good positive correlation to bacteria to the organic matter amount, with millions to hundreds of millions per gram of soil. And this is, however, these millions to hundred millions bacteria is less than 0.1% of the total mass of that soil. In water, the concentration can range from 50,000 to 50 million per milliliter, and that can depend on various conditions there. Now, another phyla is fungi. These guys you usually think of as mushrooms, but they can be all sorts of types. We're ubiquitous and diverse. They tend to favor only aerobic conditions. They want somewhat dry environments. However, they can inhabit some oxygenated pools, some lakes, some rivers, and oceans. But mostly, you see the fungi growing on land. Now, they form a wide range in form, and you can see them as large multicellular things like the mushrooms, but they can also be small forms. They'll break down our pre-synthesized carbon. They will largely chew up organic matter that is already in place and thus incorporate them into their body. And so we need a lot of fungi to help break down organic matter, to break it down into the humic material that we know more about. Now, the fungi prefer more acidic conditions to the bacteria's basic conditions. So obviously, we have where one might be thriving, the other might be a little weaker, a little bit less of them. But one thing about fungi is they can grow up to tremendous size. There's one known organism, honey mushroom, in the Malheur National Forest in Oregon, growing to about a 10 kilometer squared area. Now, if you watch X-Files episode, season 6, episode 21, they talk about it, where the whole theme evolves around this mushroom is slowly eating people by causing them to ingest the spores, and then they trip out. They start to go under some sort of mushroom-based trip, and they end up getting confused and essentially eaten by the mushroom. And very interesting. But so this mushroom is largely underground. It's not like a huge mushroom you can see from the area, but it does produce small sprouts that come from underground, so we can see the above ground structure. But it's all from the same large mushroom. Now, the number of concentration of fungi is a lot smaller than bacteria, but because they're much larger in size, they may play a bigger role. Due to the filamentous structure, they also have a very large surface to volume ratio. Now, this one I was not really particularly familiar with, the actinomycetes, which is a type of unicellular organism. Now, they have a multi-stage development where in one stage they form fine branching filaments like fungi, but on a later stage they see a population boom akin to bacteria. So they're a little interesting. They can be found on land and sea, but they prefer land with aerobic dry conditions. They prefer warm climates like tropical grasslands. So like where they can be about 30% of the microorganisms. They're sensitive to acidity, but they can tolerate higher salt concentrations than bacteria can. So it's typically more in it's probably more in South America region. So that's probably why we don't hear too much about them in the U.S. But like fungi, these are responsible for a larger amount of the degradation processes. But they can also produce natural antibiotics that can kill off and control the populations of other microorganisms. And of course, there's algae. Algae are the chlorophyll-containing organisms that can range in size from the microscopic to large plant-like structures. These are most commonly found on the upper epilimnion layer of eutrophic water, as well as moist surface layer of soil. But unlike the fungi and the actinomycetes, these produce new organic matter. They can convert the inorganic carbonate to organic carbon. So they are going to be an important part. They're just like normal plants, except they're in the micro scale. They're an important part of the global carbon cycle. And they can also usually fixate nitrogen, so convert nitrogen from the air to nitrates that they can use. Now, during these carbon conversions, other inorganic nutrients and non-nutrients need to be assimilated into their structure. And so this can be used by scientists to actually monitor environmental pollution. So you can collect some algae and study them and see how much lead or cadmium or mercury did they accidentally take up while they're building their structure. Now, algae can cope with great temperature extremes. We can find them in the polar ice or in hot springs. And finally, protozoa. These guys are the simplest form of animal life, along somewhere between 5 and 50 micrometers in size. Can be found both land and sea. In the soil, they tend to live in thin films of water on the surface of particles, and they play an important role in our microbial processes and wastewater treatment. They're going to break down a lot of our waste. But they need a lot of moisture. They need a warm climate, well oxygenated, and mild pHs. But there are some that can exist at some extreme pHs. But among other things, they can consume the bacteria and other microorganisms and thus regulate the total microbial population. So they not just eat organic matter, but they can eat our other microorganisms. So by family, that is the 5 classes. But we can also talk about them on ecological characteristics, such as autochthonious versus zymogeneous or allochthonious. And basically, whether they're an indigenous microorganism or a foreign microorganism, or essentially a foreign or a microorganism with a otherwise unrealized potential. So the autochthonious is basically here's the steady state population of microorganisms that can live and maintain a stable, happy population in the area. That some of these guys may be more active than others may be lying dormant, but the idea is that the population remains steady because there's a constant but limited food supply. The zymogeneous often is when something new comes in, such as a new bacteria that can better thrive, or it could be just a fresh input of nutrients. Like suddenly someone starts dumping phosphates into the river or into the lake, and suddenly we get all these nutrients, and the microorganisms suddenly proliferate. And so we get something burst of microorganisms, at least until the conditions start to return to normal. So this nutrients could be brought by heavy rainfall. So a large quantity of nitrate into an otherwise nutrient-poor lake would cause algae to flourish, at least until all the nitrate is consumed and then baseline conditions resume. So that's one way we can look at a population of microorganisms, whether this is the natural level versus we're seeing an elevated level. We can look at these guys in terms of carbon sources, autotrophs versus heterotrophs, whether they can produce their own organic carbon from inorganic carbonates, or they need to convert this, so whether they have to break it down from something else. Autotrophs are also called synthesizers or producers, but they can turn CO2 or hydrogen carbonate or just carbonate into the things. These are most commonly algae and bacteria are more often the autotrophic. Can be subclassified as photoautotrophs, where they need light, or chemoautotrophs, which they do chemical oxidation to do this. But so nitrosomonas are examples of chemoautotrophs in that they don't need light, they just need the oxygen to oxidize the ammonia to nitrates. Now our heteroautotrophs, degraders, are consumers. They're going to break down plant and animal residues in water and soil. Our fungi, our actinomycetes, our protozoa, and most of the bacteria are considered heterotrophs. But certain microbes are very useful for breaking down synthetic organics, such as pesticides. The Arthrobacter can break down the pesticide IPC. That when, as we can see here, that IPC, first we cleave off one side of this ammonia to make this benzoamide into an aniline, and then it's going to oxidize and convert the aniline to a diphenol, and then it's going to break up the ring and produce some CO2 and the other half of the compound. There we have like isopropyl alcohol. But so full degradation into CO2, ammonia, and other sulfates occur. This is called a mineralization. So mineralization is the process by which we convert organic carbon and materials back into inorganic, which means we're not bound to a carbon source largely. It's back in, or the carbon source is ionic and not covalent in nature. And then we can also look at and classify microorganisms by the source of electron acceptor. Are they aerobic or anaerobic? Do they need air to do the redox chemistry? Aerobic microbes use molecular oxygen either from air or oxygen dissolved in water as an electron acceptor. So, for example, oxygen plus acid produces 4 electrons. Thus, we can convert this into water. In order to be active, these microorganisms must have plentiful supply of oxygen containing air. So these aerobic microbes will be on surface waters, turbulent river or wave-covered lakes, very porous surface soil. So places where either oxygen is plentiful or oxygen is rapidly refreshed, like turbulent rivers or wave-covered lakes, it's going to be shaking things up. Or even as the oxygen gets used up, it can diffuse back like porous soil. Anaerobics is where the air exchange is limited, like deep ocean, where the oxygen has to come from the air and slowly filter down to the deep ocean. That's not very easy. Compacted soils, regions where like oxygen can't easily diffuse through the soil. So oxygen gets depleted and it stays depleted. In these conditions, anaerobic microorganisms thrive. They use other electron poor species like sulfates to induce redox chemistry, and we'll talk more about these later, but sulfate can be converted into hydrogen sulfide and a bunch of water with some electrons. Now, bacteria are the most common type of anaerobe. So we don't see a lot of, there's no really anaerobic fungi, not really any algae that are anaerobic. So bacteria is the main type. But some anaerobes are obligate anaerobes, which means oxygen is toxic. They cannot live where oxygen exists. They lack a cytochrome and a catalase that will prevent this oxygen from converting into hydrogen peroxide and thus killing the bacteria. So others are facultative anaerobes, which means they can use oxygen if it's there, but if it's not there, they can use other species. So oxygen isn't necessarily toxic, it's just not important. But so we should distinguish between what's called anoxic and oxic conditions. Where anoxic is the absence of all types of oxygen species, such as nitrates and sulfates and carbonates. So when you don't have inorganic oxygen of any type, carbonate, nitrate, sulfate, then you're in an anoxic condition, which is different from an anaerobic, which is different from an anaerobic condition, which just means there's no molecular oxygen. So we'll talk about the various species and how they work a little bit later. And one of the last ways we can classify these guys is on temperature preference or morphology, their shape. In most kinetics, microbial growth follows an Arrhenius relation. That we talked about this in Gen Chem, that the rate of growth, the natural log of the rate of growth is equal to an activation energy barrier over R and the temperature. However, there is a limit where above a maximum temperature, a particular enzyme starts to denature, and thus our growth declines. So as you can see in the graph there, you heat up, you get an optimum temperature, at which point enzymes start to die off, and so the thing can't grow. And so we can have various types of bacteria that favor different conditions. Psychrophiles, which prefer cold environments. The optimum temperature is below 20 degrees. Mesophiles, so the medium guys between 20 and 45, and thermophiles, with the optimum temperature being somewhat above 45, which would obviously favor tropical environments or say in compost heaps. So In terms of morphology, we can refer to the shape of them, whether they have a rod shape like bacilli, spherical shape cocci, or spiral shape spirula. But other morphological categories exist, but these categories don't actually help us understand their role in the environment like some of these other guys do. So the shape doesn't necessarily mean their function. While, like, if we talk about the phyla, the fungi, they're the decomposers typically. Talk about the psychrophiles. Okay, these guys exist in polar conditions, and so they're going to be active up north. Talk about how this is the native level. We can talk about, as I said, like whether it needs oxygen or not. So all this kind of breaks it down to the chemistry and less about the shape. So I'm pretty sure that's where we'll stop. That's where we'll stop, and so we'll pick up on talking about the carbon cycle on in the class. So with that in mind, hopefully we got through the basics. Thank you.